All right, folks, quiet down. Let's take out your headphones and AirPods and cell phones and all that. Yeah, I'm kidding. I had to add a little levity to this. This is so new and so weird without all of you being here. So the way this is going to work is I'm basically going to create videos that break the unit down into the subtasks that you would find in the learning targets. And to just kind of get us started on something a little bit more comfortable, I was going to pick up on subtask 2, which we've actually already covered quite a bit of if you were in class. But we didn't totally finish it. So, hey, that's a great place for us to pick up. And this is going to be page 3 and the top of page 4 in the learning target homework. So we talked about this goal, that essentially we want to be able to describe what is going on with supercoiling, which is the packing of DNA up, and the result or influence that has on gene expression, which we could also sort of describe as using the DNA. Now, we looked at the first thing that happens when you take this DNA and you pack it up, and that is you make this little structure called a nucleosome. That's like the tiniest little structure that you get when you start packing DNA together. That's going to involve 8 histone proteins, 1 H1 linker, and that is a nucleosome. And now you've got a whole bunch of nucleosomes all over. DNA is just packed up into these tiny little ball packets. So from there, you actually create a couple of other structures. The next one is a thread, and then that thread gets coiled and coiled more until you have a chromosome. So ultimately, what we want to note is the structure of a nucleosome being 8 histone proteins, an H1 linker, and then the name for that. Now, where we go from there is looking at that there are proteins wrapped up into this thread that we would call chromatin. That then wraps up into this larger structure called a chromosome, but we can subdivide that into 2 sister chromatids. Each chromatid is a strand of DNA. So there's actually 2 strands of DNA here. They are identical to one another, but one strand is on the right and its identical sister is on the left. They are connected by a centromere, and if we were to go through cell division, mitosis, one of these strands would be ripped off and sent to one cell, and the other strand would be ripped off and sent to the other cell. Now, before we get into anything else about supercoiling, there are a couple other numbers here that we want to note in the double helix. For instance, base pairs are 0.34 nanometers apart from each other. If you go through 10 of them, or 1 full spiral around the double helix, that is going to cost 3.4 nanometers of distance. So again, just noting a couple of the different sizes of things in the double helix. Now, from there, we note that the chromatin thread that they would coil into is 30 nanometers wide, and the DNA helix itself is 2 nanometers wide. Now, ultimately what we get to is this structure that is incredibly condensed. Supercoiled DNA is much, much denser and takes up a lot less space. Now, the only time that you're ever going to do this fully condensing the entire chromosome is before cell division, when you need your DNA to be easy to move and you don't actually need to be reading any of the information in it. So if you were going to be supercoiling, you would be taking all of the genes, packing them into chromosomes, and that means that none of these genes are being currently expressed. None of them are being used. They are packed up and they are unavailable to the molecular machinery that would want to be using and reading or expressing those genes. So ultimately, to sort of wrap all of this up, this little picture, I think it kind of does a nice job. This chromosome is ready for mitosis. It's ready for cell division. Again, the only time you're ever going to see full chromosomes is right before a cell divides. And again, that's because right before a cell divides, it needs its DNA easy to move around, packed up, but it doesn't need the DNA to be used. So none of these genes are being expressed right now. This chromosome is what a chromosome would normally look like. It doesn't really look like a chromosome at all. But all that DNA is stringy, it's unpacked, it's not wrapped up around histone proteins, it's not packed into nucleosomes, at least not fully. Not all cells are going to unpack every single gene, because no cell uses every single gene. So for instance, if this was inside of a pancreas, there would still be a little area that's packed up and that would be some of the genes that the pancreas isn't using. But then there would be other genes that are unpacked, that are expressed, that are not supercoiled, and that might be something unique to a pancreas, like creating the protein insulin. You have to read the insulin gene if you want to make insulin. So coming back to our original goal, the relationship between supercoiling and gene expression is that if you are doing one, you are not doing the other. If you are supercoiled, then you can't express the gene. You can't use it, you can't read it. You would say that it is wrapped up around histone proteins. You would say that that gene could not be transcribed. The inverse is also true. If your DNA is not supercoiled, then you can use it. You can express that DNA. It would not be wrapped around histone proteins, and you would be able to transcribe it. Now, that information should be enough that you can take a shot at any of the information in subtask 2. Click on the next video for subtask 3.